He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
So we waded out a bit deeper to have a look, and you're using the bathoscope to look at the bottom.
And all I can see is clams. Clam siphons, clam shells. So even if it looks like gravel, you look a bit more closely and you can see these little holes, which are essentially the siphons. These little apertures that they use to feed and inhale.
Yeah, when you look down first, it just looks like kind of muddy gravel, and then you see it's got this like pumice texture, and you've explained that those holes are actually the siphons.
That's right.
So it's just clams all the way down.
Clams everywhere.
Kia ora, nau mai haramai ki te ao hurahanga. Hello and welcome to Our Changing World. Ko Claire Kincannon tēnei. Dr. Michelle Melchior and I are at the end of a little jetty at Bob's Landing in Lake Karapiro on the Waikato River. We're dressed in waders and holding bathoscopes, a traffic cone-like piece of equipment with handles and a clear bottom, so you can view into the lake. And at our feet, all around, are invasive gold clams.
What we've been finding in some spots is that the number of clams can exceed thousands per square metre.
Michelle is a freshwater scientist at Earth Sciences New Zealand in Kirikiriroa, Hamilton. And she's part of a big collaborative programme involving Mana Whenua, Council, the local university and international partners. It's aimed at understanding these invasive clams, trying to predict what might happen next, and developing different strategies to manage them and to prevent their spread. The clams, Corbicula fluminea to give them their scientific name, are native to Southeast Asia, but they've made their way to lakes and slow-moving rivers in other parts of the world, including North and South America and Europe, where they've spread rapidly. In 2023, some freshwater ecologists spotted the small tan-brown shells here at Bob's Landing, just off State Highway 1, and alerted the Ministry for Primary Industries, or MPI. We don't know how they got to New Zealand, but it's likely they'd been here for several years already. A subsequent survey to investigate how far the clams had spread started at Lake Taupo and worked backwards. It revealed that they've made their way as far inland as Lake Maraetai by Mangakino and pretty much all the way down to where the Waikato River enters the ocean at Port Waikato. There was a second clam species, Corbicula australis, that showed up in a small man-made lagoon at the Lake Taupo Aquapark in March 2024. Luckily, because it was fully contained, that species was easy to get rid of. But the spread of Corbicula fluminea along the Waikato fits with what's been seen overseas. These clams tend to take over, and once that's happened, they've not successfully been eradicated from anywhere. There are a few reasons why they're such successful invaders, but the main one is how they reproduce.
We just need a single clam to reproduce, so it's a hermaphrodite, self-fertilising, self-reproducing. So that means that one clam can produce 70 to 100,000 babies annually.
Sorry, 70 to 100,000?
Yes.
In 1 year?
In 1 year. So thinking of all of these clams that we have here, they can release lots and lots of little babies into the water column, and those babies are called veligers. And they're microscopic in size, so they're around 200 microns, so 0.2 millimetres in size when they're first released. And so we can't really see that they are in the water. And so that's the issue, right? So those little babies, they get kind of dispersed wherever the flow takes them. And so if they're being dispersed into water intakes, piping of power plants, for example, no one's going to know that they're there until they've started to grow, and then that's when they start clogging. So then another problem is then their juvenile stage. That stage produces a byssal thread, and that byssal thread attaches onto surfaces. So if there's a leaf floating around, that can then help it disperse to different places as well. Or if you go into the waterway in our waders, for example, you know, they may kind of attach onto your waders or onto any kind of gear that you have, and then you can if you're going into another waterway, that can then disperse that way, so overland, transport into another waterway.
And it's all about to kick off again, says Michelle, as the water warms. When it reaches over 15 or 16 degrees, the clams will begin reproducing and releasing larvae. They've got two siphons, essentially an intake and an outflow, which they use to filter the water to get the bacteria and microalgae they feed on, and to excrete what they don't eat. And the larvae will also get spat out over the outflow siphon. All their basic biology has been well studied overseas. Along with the siphons, they've got a foot that they use to move around and bury into the sediment, which they can also feed through. To understand how they work in a New Zealand context, the team have been doing annual surveys at sites in Lake Maraetai and Karapiro.
That is to essentially understand densities at different sites and different habitats, to better understand what the population is doing over time. Are they establishing? Are they not really liking the habitats in certain areas? Are they growing in population? And so, we take these nets. We usually have a quadrat that's laid out underneath the net, and then we use this net to scoop up the substrates. So we go to about 10 centimetre substrate depth, or try to at least.
And so you're just kind of jamming the metal bottom of the net in. Right, you've just listed up a handful of, I want to say gravel, but actually it's mostly clams rather than gravel. For your survey then, do you go through and count each one of the clams that you find?
So we count each of the clams, we measure them to better understand size distribution, to see if there's any recruitment happening, which there is.
Recruitment means small new baby clams coming in.
Small new baby clams, lots of reproduction happening. We're also finding populations with older individuals. So these ones look like they are around 25 millimetres, so 2.5 centimetres in length. But we are starting to find larger clams, so around 4 centimetres, which means that they've been here for a while, so a couple of years.
Do we know how long they live for?
There's research out there that suggests they live around 5 years, so they reproduce in high numbers, grow super quickly, and then die really quickly. So it's just an ongoing cycle of lots of reproduction, lots of small babies everywhere, growing in large numbers, high densities, and then dying.
It's an absolute winner of an evolutionary strategy for the clams. Meanwhile, water infrastructure is one loser. The clams jam up power and irrigation companies' equipment. And who else loses out? With so many clams, what happens to the lake and to the native species that also live there? While she was doing her master's research about 7 years ago, Michelle used to come here to Bob's Landing to collect some of our native freshwater mussels, kākahi, for study.
And it would take us sometimes hours to collect 100 or so kākahi, and we would use these stethoscopes to find what looked like a kākahi in and amongst the gravel, pick them out, put them into a bucket, and take them back for research. Now with the clams, it's a completely different story. So we don't need these stethoscopes anymore. We can even just take a handful into the substrate, and what we'll have is 5 clams. And so now what we use is this little clam net where we can just scoop up sediment, and amongst that is 50 or so clams, and we just need to take a couple of different scoops and we'll have hundreds. So the contrast between a couple of scoops in a couple of minutes and hours to find kākahi is it's been very confronting, especially because kākahi are a threatened species. So Caradella menziesii is the species that lives within the Waikato River.
And are you still finding kākahi here?
We are. So last week I went out to do a kākahi collection, but again, there are very few in and amongst thousands of clams. So they still exist here, but they are kind of being overcrowded and overrun.
And do we know... I mean, it seems intuitive that if you have golden clams that are in this kind of density, then they're just going to take over the habitat. They're just going to take over the food, and it's going to be bad news for the kakahi. But do we know that for sure?
There is some research overseas that suggests that they are out-competing, so the clams can out-compete them in terms of food, but also in terms of reproduction. So they have found that clams in high densities can suck up Glaucidia, so kakahi larvae, which then, reduces their reproductive output as well. So those kinds of things. In some locations, kakahi can withstand the clam densities and the clam invasion, but so we don't know for sure, and we don't know how the clam will behave in New Zealand waters. It's early days, so we're literally just witnessing the early stages of the invasion.
Yeah, I mean, you say early stages of the invasion, but it's hard to see how they could get any denser in a place like this, where they are kind of already everywhere.
So we are seeing them now in 500 to thousands of clams per square metre. Overseas, they're finding them in tens of thousands of clams per square metre. So they're on top of one another within an area like this, which we don't want it to get to that stage. It'll be devastating if it does.
So for now, you're just trying to collect some gold clams for experiments back in the lab?
Yep, that's right.
How many do you need?
We need a couple of hundred, but that's easily done.
Depressingly easily done.
Exactly. We might even have 100 in this sieve right now.
Oh, I see. You've got a layer of sieves.
Yep, nested sieves. So some larger mesh sizes, followed by small, very small, just to collect all kinds of size classes. We are wanting to use these clams for benthic barrier trials. So benthic barriers, which is essentially matting on top of the lake floor, has been one of the ways that's been quite successful at killing clams overseas. So overseas studies have only done it within lakes using rubber matting, and what we are wanting to do is start off with small scale trials, so within the laboratory, and compare that rubber matting with something else that we could potentially use in New Zealand, whether there's something that's more efficient that can reduce the kind of kill time, because it currently takes around 35 days using benthic matting to kill the clams. So if there's something out there that can reduce that time, that would be really, really great. So we're just starting to run trials on that.
So you were saying it's been shown to kind of work overseas. Where is that? Is that North America?
North America, Lake George and Lake Tahoe, they have undertaken benthic barrier matting surveys. So those are huge, I think 9 metre wide long mats.
So they're literally just putting out these rubber mats onto the bottom of lakes to try and kill the clam.
Mm-hmm, that's right.
Does that not just take out anything?
Yeah, so what we're also doing as part of this research program is looking at anything that we're doing with the clam, any kind of intervention, what will that do to non-target species? So we're looking at kakahi, we're looking at koda, we're looking at potential fish species like lamprey that can burrow in the substrates. So anything that we're doing, we want to know that it is not going to impact our native taonga species as well.
It's hard to see how it wouldn't if it was taking out the oxygen.
Clams are very intolerant of low oxygen. We don't know what kakahi will be like in comparison. So if we can find a sweet spot where we can kill clams but still are able to have kakahi that stay alive, then that would be great. Otherwise, we might have to come up with other strategies, so potential kakahi translocation prior to the addition.
Remove the kakahi from the area and then put down the benthic barriers to effectively smother the clams.
That's right, yeah.
You can probably hear in my voice that I'm a bit overwhelmed. I'd known some facts about gold clam reproduction, but to see so many of them in the lake and then to hear that it might get even worse has sort of floored me. That, plus the fact that people overseas have been grappling with this for a lot longer than New Zealand, but that the best solution to date to manage them, not even to eradicate them, is to smother them with rubber mats? With that in mind, preventing them from spreading further is crucial. So alongside the benthic barrier trials, Michelle and the team are also trying to figure out the quickest, easiest ways to ensure that gear used around water is clam-free. Right now, there are specific MPI check, clean, dry rules for the North Island, based on results from an earlier round of trials on how to kill 2-day-old juvenile clams. Check means remove anything visible. Clean is to get those invisible things, not just a rinse off, but by soaking in hot water, bleach solution, or salt water, or freezing the gear overnight. And then fully dry out everything for at least 2 days before using again. On Earth Sciences New Zealand's campus, they have a giant walk-in freezer. That's their go-to method for decontaminating gear. But that's not something most people just have at their home. So they're testing methods and times that will be able to kill clams of all age ranges. With the clams in a bucket, we climb into the ute and head back towards the city. Gold clams have been given the legal status of an unwanted organism under the Biosecurity Act, which means that people must not knowingly move them or any water that may contain them. But Michelle and the team have a permit to allow them to collect and work with the clams.
So this is one of the rooms that we're using for clam trials.
It's a small room with, as you can hear, a loud fan whirring. The 2 lab benches on either side have different experiments set up on them. On the right-hand side, a stacked array of Perspex trays has shot glass-shaped containers, each with some sand and a clam inside. They're arranged by clam sizes and have been treated with different conditions to see what might kill them, including one labelled harakeke leachate.
So another part of our benthic barrier trials is to test different types of natural materials that we find in New Zealand. And so one of the things that has been suggested is using harakeke matting alongside of potentially rubber matting to kill the clams to reduce the period that we leave that matting down to kill the clams. And so we're currently testing what comes out of that harakeke when it's been left in water for a while. So that's the leachate. So we have left harakeke in water, we have filtered that water that the harakeke has been left in, and now we are testing that water on the clams to see whether that has an effect on the clams.
And then what's going on over here?
We're just housing different clams for future experiments here. So we're running substrate trials as well in the future. So substrate preference trials. Do they like sandier substrates? Do they like more gravelly substrates? And that'll help us determine what we can do in terms of predicting where they might go in the future, where they might spread to, what areas they might like to thrive in and establish.
This is also where they'll do the initial small-scale rubber matting barrier trials. And they've got another gold clam management idea that they want to investigate too.
We're in the early stages of looking at how acoustics, so different types of sound frequencies, might be used as a deterrent or as an attractant for different valiga or juvenile stages of the gold clam.
And that's based on evidence that has been seen in the marine environment, right?
That's exactly right, yep. So there's some evidence out there that certain sound frequencies or noises can disrupt the byssal thread, so the thread that allows clams to biofoul. There's also been some evidence that has shown that it can disrupt different life stages or the development of different stages. But there's also other research out there that has shown that certain sound frequencies might attract biofouling species at early stages to different areas. So we're going to test a whole bunch of different sound frequencies to see what we could potentially do with the valiga stages and the early juvenile stages of the clam.
On the opposite bench sits a complex-looking setup, with a single clam each in a little box that has pipes leading in and out of it. Freshwater scientist Carl Safi talks me through it.
This experiment is really focused on their impacts, so it's how they're affecting the natural system. This is a preliminary study. We've collected water from the river and brought it here, and we're keeping everything contained. And so we're setting up a series of chambers here which have individual clams in them, and also got some control chambers which have control clams, which are actually just a shell with a weight inside them to keep them sitting down in the same position. And we're flowing water through these chambers, which are baffled, at a constant rate, with the idea that we can see what the clams graze on over a period of time, and then we can back calculate how that will affect the populations in the lake.
Right, so you're going to investigate what's in the water before you flow it in. You know exactly the rate that it's going past the clam at, and then you're going to investigate what's in these little buckets that are the other side of the clam.
That's correct, yeah. So we're looking at how they impact different populations, so specifically looking at phytoplankton, but also looking at how they might graze like detrital particles and other things that are in the water. And we can look at different size classes of those organisms. Like in this case, it will be whether they're grazing the very small plankton, like picos, which are less than 2 in size, or whether they're grazing larger plankton, and whether there's a preference between the two. We also collect off their siphons feces and pseudofeces, and feces are what they actually ingest through their gut and pass out, and pseudofeces are what they take into their mouths and then go, oh, I don't like that, and spit it back out. So we measure what's in those two collections to see what they actually ingest and use to build their tissue and what they might reject.
With all of this, you mightn't be too concerned if there were only a couple of clams. But the sheer number of them means they're likely having a significant impact on the lake's water chemistry and the makeup of the tiny critters that float in the water. Something that boat users on Lake Karapiro have noticed and mentioned to Michelle is that the water is clearer. This hydro lake is much modified. There are invasive pest fish and weeds and high sedimentation. But the action of the huge number of clams filtering the water appears to be improving the clarity. However, this, unfortunately, does not necessarily mean that the water quality is being improved. That's why the team are also doing quarterly monitoring of the water's microscopic critters and chemistry.
We are measuring a whole lot of variables in the water column to understand how that may be changing with time, because if they're removing, as I say, food that affects other organisms, we want to know how that population structure is being affected and understand how the particular characteristics of those lake systems might be impacted by their presence. Yeah.
When you say population structure there, you're talking about the small little things that are floating in the water.
Yeah, well, I suppose the way you've got to look at it is the small little things provide food for everything. So it's that whole food chain process. So really the very small algae are very important to the system. And the zooplankton, of course, consume those. So if their population is being removed, then our zooplankton won't be around, and then you'll get impacts on things like fish populations as well. So we're looking at the whole food chain effects, and of course on native species of mussels or clams that will be affected as well. Yeah.
And alongside that, with the monitoring, are you also looking at changes to the chemistry of the water?
Yeah, we look at all types of variables, so our nutrients, temperature, things like carbon, just general mass of biologicals in the water. So we're looking at all those sorts of things, and obviously this could have an impact on our sediments, because the behaviour of these clams, they might disturb the sediment, causing more nutrient release, etc. So there's a whole lot of processes being studied.
Overseas, it's been reported that in high numbers, the clams can release enough phosphorus and nitrogen to reduce the amount of oxygen in lakes or increase algal growth, both of which tend not to be good for other lake life. The mission of the programme, to understand the clams, predict what comes next, and try to minimise their impact, continues. In the meantime, the gold clams are here, and they're multiplying. Michelle says the best thing we can do is not let them spread any further.
Checking your gear after you've left the Waikato River system, cleaning, is really, really important, because microscopic things you can't see. So making sure that you clean anything that has been in the water, and then making sure that the gear that you use is completely dry is really, really important.
Thanks to Dr. Michelle Melchior and Carl Safi of Earth Sciences New Zealand in Kirikiriroa, Hamilton. This episode was produced by me, Claire Kincannon, and sound engineering was by Phil Bench. Editing was by Kate Newton. Learn more at rnz.co.nz/OCW. And if you have any comments or questions, you can email ourchangingworld@rnz.co.nz. We also have a monthly Our Changing World newsletter. You can find a link to subscribe to that in any of the episode details on the RNZ podcast page or at the bottom of any of the web articles at rnz.co.nz/OCW. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, the wiki.